very good evening, ladies and gentlemen. I wish you a very warm welcome to Monash University, Malaysia, if you're from outside, and everyone to the 4th Sir John Monash Lecture this year. The Sir John Monash Public Lecture Series provides Monash University, Malaysia an opportunity to engage the community in Malaysia and the region through the promotion of thought leadership in key areas that are strategic to the development of the region in the 21st century. This lecture series takes inspiration from Sir John Monash, one of Australia's most distinguished citizens, a scholar, a leader, and a man who sought to use his education and abilities for the benefit of the community. The public lecture series covers a wide variety of contemporary and interdisciplinary topics presented by high-profile thinkers, international leaders, policymakers, corporate leaders, and leading scholars. All are distinguished in their areas of expertise and whose work significantly impact the global community. This evening, we are delighted to host Professor Dr. Mark Seielstad from the University of California, San Francisco, to deliver his lecture entitled Nature and Nurture in the Growing Diabetes Epidemic. This lecture could not be more timely. Over the past 10 years, there has been unprecedented rise in the incidence of diabetes in Malaysia, and we are grappling now with an epidemic where nearly 1 in 6 adults are affected, and the disease is alarmingly on the rise in children. Before we commence the lecture, permit me to briefly introduce our speaker. Rest assured, I will not be reading 45 pages of his CV, and I'll go to a brief introduction. Professor Dr. Mark Seielstad was educated at Stanford University, receiving 2 simultaneous bachelor's degrees, one in biological sciences with honors, and the other in classical studies. He then completed a PhD in biology at Harvard University and joined the faculty at Harvard University School of Public Health. In 2002, he worked at the Genome Institute of Singapore as group leader for population genetics and rose to associate director in 2009. Shortly after, he moved to the University of California, San Francisco, where he is currently professor in genetics, epidemiology, and biostatistics. Mark's research focuses on the identification of genetic variation contributing to complex diseases like type 2 diabetes and autoimmunity. His illustrious career has been spent learning, teaching, and researching in many countries, including France, Ethiopia, Sudan, Greece, Italy, Thailand, Indonesia, and Malaysia. He has several active collaborations in this region, and I would vouch to say that he is happy to be back. Ladies and gentlemen, please join me in welcoming Professor Mark Seielstad to deliver the lecture.
Thank you very much for coming, and thank you, Ma, for that kind introduction. Thank you for the invitation to visit your university, which I was fortunate enough to visit several years ago. It's always a pleasure to come back to this part of the world. As Maud mentioned, I did work in Singapore for 8 years, and it feels a bit like coming home. So I'm divided my talk into 4 little topics that I hope will be easily metabolized or digested. And first, I will just give you a brief introduction about what is diabetes and why it's important. Then a brief introduction, 10 minutes, everything you need to know about genetics, and try to summarize some of the findings that genetics, some of the insights that we've gained into the diabetes disease process through the study of genetics. And then end with a discussion of 2 new areas. One, epigenetics, which is really at the interface between genes and the environment. I'll explain more about what that is, but it's an instance in which our own DNA can be chemically modified by certain exposures in the environment, affecting how the genes are actually read out and expressed. And so it enables very rapid adaptation, potentially, to aspects of the environment. And then I'll end finally with a discussion of the gut microbiome, which entails this vast microbial community that resides within our gut and helps digest our food, and it really is very centrally involved in our metabolism. as a result. So these are two new emerging areas that I think will turn out to be very important. And getting back at this theme of nature versus nurture, which is a little bit of an old-fashioned way to think of it, but how much are we able to blame our parents for our poor health outcomes and the way our life turned out through the genes that they've given us, and how much responsibility do we need to accept ourselves for the poor lifestyle choices we might have made, our poor diet and sedentary lifestyle and things like that. And of course, the good news is there's blame for everyone to go around, because it's really very complex, and genes and the environment are both very important in that. So as you probably know, the sugar glucose is really the primary cellular energy source. It's the source of energy that's available and usable by all cells, and that even other nutrients like fat and proteins, when they're broken down for energy, end up being converted biochemically within our body to the sugar glucose, which is really what's used in all cellular processes. When we eat, after we eat, there's often a spike in sugars, as you might expect, and our body deals with that by releasing a protein called insulin that's produced in the pancreas. And that protein signals to the peripheral tissues, the fat deposits and skeletal muscle and so on, to absorb the glucose, to maintain a sort of even keel, so that glucose is taken up and stored in times of abundance and then released in the hours after a meal, so that the sugar is always available at basically the same level, and it doesn't rise to super high levels. And so diabetes is an aberrant state caused either by the lack of insulin or an insensitivity to it, perhaps due to chronic overexposure, and it leads to chronic condition of high blood sugar. And that's really how it's diagnosed, is by measuring blood sugar, fasting blood sugar, and seeing that it's higher than it should be. There are two main modifiable risk factors: sedentism, lack of exercise, and overeating and obesity. And it's thought that those can actually end up exhausting the pancreas, so that eventually it gives out and stops producing insulin, and as well as making the peripheral tissues desensitized to it. And the complications over a period of time are actually quite serious and severe. Although it takes a long time to develop, most cardiovascular conditions, heart attack and stroke, actually have diabetes as an underlying cause, and there are a lot of microvascular complications as well, including retinopathy, which can lead to blindness, nephropathy, which is the main indication for dialysis and kidney transplant, and then peripheral nerve pain, and eventually insensitivity and poor wound healing, leading to amputations in the peripheral limbs, especially the feet and legs. There are several major forms of diabetes. There's type 1 diabetes, which is actually an autoimmune disease, where your own immune system, which is meant to defend you against infections, aberrantly recognizes the insulin-producing cells of the pancreas and attacks them, destroys them, and leads to absence of insulin. It's usually of juvenile onset, and it's historically been called insulin-dependent diabetes because it can be easily, well, perhaps not easily, but it can be well managed by glucose sugar monitoring and the administration of insulin injections. That's about 5% of cases globally, and more common in Europe, not so common in Asia, it turns out. But the predominant form, and most of what I'll talk about today, is type 2 diabetes, which encompasses 90% of cases. It used to be called adult onset, but as Maud intimated in the introduction, because obesity and sedentism are occurring much earlier in life, there are now instances of juvenile onset type 2 diabetes. It used to be called non-insulin dependent diabetes because it was thought that it wasn't a matter of insulin abundance, it was just the body's inability to respond appropriately to the insulin that was being secreted. But, and partly it's the result of genetic findings which have implicated insulin secretion and production. It's now appreciated that insulin can be a useful therapy even for type 2 diabetes. Then there are various sort of single gene Mendelian forms of disease that go under various categories, and those have provided very nice insights, but aren't very important on the population scale. So we tend to think of diabetes as a problem of the developed world, but it turns out you find it everywhere, including sub-Saharan Africa at very high levels. So more than now today, 400 million people alive today are experiencing diabetes. And 80% of them live in low and middle income countries, which we think of, we don't often think of chronic diseases as being important in these parts of the world, but it really is. And we need to add that to our understanding of the health issues in those countries and not just focus on the infectious disease issues that we're all very familiar with. A lot of diabetes goes undiagnosed, and so there can be years of chronic damage that build up before it actually manifests, and at that point it's often very difficult to reverse the symptoms or to have effective therapies. And of course it's worse in the less developed parts of the world, but in most countries there's a lot of undiagnosed diabetes. And we tend to think of it as a disease of old age, but it turns out that at least half of people who die of diabetes-related causes are under the age of 60. So this, we probably all know people, maybe even in our families, but certainly friends and so on, who have people affected by this disease. And we often, the symptoms aren't always overt and obvious, but over time, it's really a very devastating disease. Now here in Malaysia, this is data courtesy of Dr. Faisal Izwan Mustafa, who I met when I was here a year ago, and I think I spied him here in the audience. There he is, yes, today. So thank you for coming again, by the way. And you're at the Ministry of Health, I believe, or in any case have done population surveys over the years here in Malaysia, and has documented really sharp rises in the prevalence of diabetes and also in the rate of undiagnosed diabetes here. So just from 2006 to 2011, a 5-year period, it went from 11.6% prevalence to 15.2%. And a lot of that is due to, just here in green, the undiagnosed disease. And so this is something that you really want to pay very close attention to. Here again, some of the shocking numbers that Maud referred to. Here by age is the prevalence of diabetes from 1996 and 2006 and 2011. Very sharp rise, so that more than a third of individuals over the age of 60 today in Malaysia are apparently affected by diabetes, or certainly problems of prediabetes and insulin insensitivity. And in Malaysia, it's even an outlier amongst your neighbors here in the ASEAN world. So this is quite a significant issue here. And if for no other reason, it's costing more and more money. But that should get people's attention, right? So I think the estimates are that at least 16% of the national healthcare budget here are spent on diabetes care. It's probably perhaps more than that. And so Malaysia is really here in the ranks of these very small island nations where diabetes is a known problem, and the Gulf Arab states as well. So there's certainly a good reason to be interested in diabetes. So as my title suggests, diabetes, of course, is related. It has both genetic causes as well as environmental ones. Genetics is usually studied using estimates of heritability, looking at identical twins, for example, and asking how often, if one twin has diabetes, how often is the other twin affected by diabetes, and comparing identical twins who are 100% genetically identical with non-identical twins who are on average 50% genetically identical, and asking, is that concordance rate higher for the monozygotic twins than the non-identical twins? And that the difference between those two concordance rates gives you some estimate of how much of the trait is due to genetics. And so for diabetes, it's estimated that about a third of an individual's total risk of developing diabetes is due to the genetic variations that they may have inherited. And so that's substantial, and that's a lot, but of course it means that 2/3 of your risk are due to lifestyle and other environmental things that aren't very well understood yet. And of course, the acid test is that to date now more than 100 genes have been implicated in diabetes using genetic methods. So that does suggest the heritability is at least somewhat accurate. But it's certainly not the whole story. So now here's the introduction to the genetics, I promise, and I hope I'm targeting this right. It's very hard to know at a public lecture, who's here and how much genetics you know. So don't be offended if this seems elementary, but I do think it's maybe a little bit useful. And like I say, this is really all you need to know. It's not, don't make it more complicated than it is. So we know that genes are made of a molecule called DNA, and that's a long polymer made up of 4 different constituent types, which we abbreviate A, C, G, and T. So it's just a series of these in often random order, but in genes, the sequence of those letters actually codes for the sequence of the proteins that are going to be made by that gene. So a gene produces a protein, and those proteins perform all of the activities that are necessary for the cell, all of the biochemical reactions, all of its structural aspects, all of the sensors and molecules on its surface. that sense the environment and everything else. Those DNA molecules are packaged into things called chromosomes that are wrapped very tightly and are in the nucleus here. Now, we humans have only about 22,000 genes that code for proteins. That is somehow a small number, it seemed to many people. There's a lot of additional diversity that can be made from those 22,000 genes, but it's just 22,000 genes. They're arranged on 23 chromosomes. We have each of our cells, more or less, in their nucleus has 46 chromosomes, one of each pair we've received from our mother and one we've received from our father. Our whole genome is about 3 billion nucleotides long, or 3,000 million, maybe you say here. And those of you old enough to remember the Human Genome Project, that was a project conducted in the 90s, a large international consortium that cost several $billion. And that was just to determine the nucleotide sequence of a stereotypical or reference human for all those 3,000 nucleotides, 3 billion nucleotides. And it allowed us to identify the genes and their sequences and what they might, what their amino acid sequences might be. So that gave us one single reference. But genetics is really the study of variation. The question we ask is what differences between me and you or you and your friends are due to genetic differences. And so that variation arises through a process of mutation in each generation as the DNA gets copied. Mistakes are made, even though not very frequently. Maybe less than 1 in a million base pairs, a mistake is made that introduces a new sequence in a particular position. But over time, variation will accumulate in populations. And so while each of us is 99% similar, regardless of race, ethnicity, or anything else, actually, that does sound very similar, multiplied over a genome that's 3 billion nucleotides long, that's actually several million differences between any one of us. A lot of that probably has no or very little significance, but some of it does encode for differences that do give rise to the apparent differences between us or our risks of disease and things like that. So after the completion of the Human Genome Project, beginning in the early 2000s, the focus was on identifying and cataloging variation in human populations. And that work proceeded very rapidly. It turns out, as you might expect, the first kind of variation that you would find is the common variation that's frequent in the population. It's just like anything when you're searching for something. The first thing you find, just because the very fact you found it, it suggests that it's probably fairly common in the population. So these are variations that are between 5 and 50% frequency in the population. So when I'm talking about variants or alleles, I'm talking about 2 different potential nucleotide sequences at a particular site in the genome. Some people might have a G, other people might have an A. And that A allele might be 5–10% frequency in the population. The G would be 90 or 95% frequent in the population. And rare variation, as we'll get to at the very end, is a much more difficult kind of variation to find, and we're only now able to do it in a cost-effective manner. So at the same time this variation was being identified and cataloged, technological developments occurred that really piggybacked on the developments in the computer chip industry that allowed miniaturization on the same kinds of silicon chips that are used in computer design and transistor circuits, that it enabled the miniaturization of DNA sequencing reactions. So the technical details don't matter to us. Long story short, you can determine the nucleotide sequence in an individual DNA sample for 500,000, a million, 2 million SNPs, or positions in the genome that are variable. And you can do that in a very cost-effective manner. So you put a sample of DNA on this, and you know at any one of various positions what the nucleotide sequence is for any particular individual. And the approach that's been successful in complex disease genetics is something called the Genomewide association study, which is typically a case control design. Very simple. You just enroll a bunch of patients with a particular disease, and then a bunch of healthy people from the same population who are otherwise similar but do not have the disease, and then ask for each one of those 100,000 or million variant sites, is one of the alleles more common in the people with the disease than in the healthy controls, more or less common? And when the frequency is elevated, that suggests that that variant or something very close by to it is playing a role in increasing or decreasing the risk of disease. So here's just a stereotype, a sort of cartoon example, but it was one of the earliest instances in which this was successful, looking at patients with Alzheimer's disease and a particular gene called ApoE, where this 4 allele, one difference in the DNA sequence is found in about 40% of patients and only about 15% of the general population. And that's actually a fairly big effect. But it means that 60% of people with Alzheimer's disease don't have that variant at all, and 15% of healthy people do as well. So inheritance of these genes only slightly increases your risk of developing the disease, but there are lots of additional factors that play into this. Many different genes and many different environmental exposures. This is not important. To be successful, you need large sample sizes, basically, and that's now been done for type 2 diabetes. And this graph summarizes really up to the minute. This is a review that's coming out this month in Nature Reviews Genetics, which just shows the progress in discovery of genes associated with diabetes using this genome-wide association approach. And this is by year of discovery, and it plots each study published in those various years where the position of the circle is proportional, and its diameter is actually proportional to the sample size of that study. And then here is the list of genes that have been identified in each of these studies. This number is now more than 120 different genes. And you see this started out very small. Things were expensive. People had very small numbers of samples. And success was fairly modest. But as sample sizes increased, discovery rapidly ensued. And there are now hundreds of thousands of samples that have been used in this kind of research. Notice, and the color refers to the population of origin. So purple is populations of European origins, and most of the studies, as you might expect, have been done in such populations from Europe or North America. But this turquoise color, there are a few studies that were done in East Asian populations, and some of these are studies that I was involved in Singapore, and some in South Asians as well in green, and African Americans and Latinos following up. But the point is, there's a lot of additional work to be done in populations of non-European origins, even though, as I'll show in a minute, it may not yield a lot of new discoveries. So I can't, and you wouldn't want me to go through that entire list of 120 genes and talk about them one by one, I'm sure. So just a few sort of high-level insights that has provided. First, keep in mind that most of these genes, we have no idea what they do. I told you there are 22,000 genes, but the dirty little secret is the biology community only knows the function, understands the function crudely in many cases of just 4,000 or 5,000 of those. And 10, 15,000 of them are completely unknown. We really have no clue at all what they're doing. So that's number one. And the good news is that if we identify genes through an association with a disease, we have a clue about what they might be doing, and then we have the motivation about really doing the hard work of the experiments to try to understand them in more functional detail. So that's number one. But for the genes whose function we do understand, they can be grouped into several different categories. On this side, these genes here, this family of genes really, are associated with reduced beta cell mass. And this other set of genes is associated with beta cell dysfunction, and that results in reduced insulin secretion. So remember, I told you that this used to be called non-insulin dependent diabetes, and that's because it was thought that insulin levels were not an issue. But it turns out this suggests that it could be, and as I say, endocrinologists are increasingly turning to insulin shots and so on to treat type 2 diabetes as other treatments lag and fail, or even at early stages of disease. Now, the other genes fall into pathways that can be roughly correspond with insulin resistance, either through obesity or through other mechanisms. So in a way, this tells us what we kind of knew already, except it does indicate that we should focus more attention on the insulin production side as well. And another major insight is that some of the existing classes of drugs that are used to treat diabetes are actually target some of these genes. So there's a class of genes called sulfonylureas. They affect the action of KCNJ11 and TCF7L2. And there's another group of class of drugs called thiazolidinones. I know there's some pharmacy students here or faculty in the audience, and I'm sorry to butcher that. But anyway, they actually act to increase the activity of a gene called PPAR gamma. So that's kind of interesting, and what it really suggests is that all the other genes, all these other 120 genes, are very likely to be productive targets for new therapies and new drugs. And so I think pharmaceutical companies are now just beginning to embark on drug development programs that are centered on human genes. They had really been wedded to this idea of using animal models, and that really hasn't panned out very well. So I think this is a productive change in emphasis. Now, I'd just like to talk about something called the Asian diabetes paradox. So as you probably know or suspect, the Americans, and maybe Australians are quickly catching up, are by far the most obese and overweight country in the world. So overweight here in light green and obese in dark green. More than 60% are either overweight or obese, that being a major risk factor for type 2 diabetes. Of course, in Asian populations, those prevalences are much lower. Very few overtly obese people by this traditional clinical standard of body mass index. And yet, rates of diabetes are at least as high and sometimes higher. And that's led to something called the Asian diabetes paradox, which has suggested to some people that maybe there are genetic differences between Asian and European populations that increase the risk of type 2 diabetes, even pound for pound, if you like. And so this is just a brief summary of work that we did in a variety of Asian populations a couple of years ago now, where we identified a number of genes here. These are just the p-values, essentially, of all of the variant sites that we looked at across each of the chromosomes. The higher the plot here, the more significant the p-value. So up here, about 10 to the minus 15. Very, very small p-values indicating that these genes are very strongly associated with the disease. And long story short, for these common variations that are common in the population, we don't see much heterogeneity between Europeans and Asians, suggesting that at least these common variants are not the source of the differing diabetes risk based on BMI. We've enlarged these studies further now with 26,000 cases and 83,000 controls. And again, the details here aren't important, but we see even less, frankly, heterogeneity between the associations in these populations. So for these common variants at these genes, we see very little evidence of genetic differences. And it kind of makes sense. If an allele is common in one population, because of our recent shared history, evolutionary history, if you like, the variation that's common in one population is basically shared across all populations. So summary of findings to date from the GWAS era are that most variants being common are shared across populations and have a modest impact on diabetes risk. If an allele is 50% frequent in the population and the disease is only 10% in the population, you know that gene is not a strictly causative gene. It might tilt someone in favor of developing diabetes, particularly in conjunction with other environmental exposures. We just don't know. But that's one way of looking at it. We understand better that insulin secretion and production are much more relevant in type 2 diabetes than we thought, just based on the physiology of the genes that have been discovered. And multiple genes that have been implicated in GWAS are targets of existing drugs. And so we presume that we'll be able to develop many new therapies targeting additional genes that are discovered. So that's sort of the glass half full. There's also a narrative that this hasn't been very successful. So that this is a fairly out-of-date graph now, but it hasn't actually changed very much. So for a variety of complex diseases here, when this number of genes were found to be associated, and with type 2 diabetes it was only when there were 20, but this number hasn't changed very much. Those 20 genes only explained about 6% of the total heritability, and yet I told you it's 30% due to genetics. So there's at least 24% of relevant genetic variation that has yet to be identified. And in fact, one of the disappointments has been that all these genetic discoveries haven't improved diagnostics very much. So this just shows the sensitivity and specificity of predicting whether someone will get diabetes using traditional clinical measures or family history, and then doing that in conjunction with genotypes at additional loci that are known to be involved in type 2 diabetes. And you can see that there's almost no change here. So adding the genetic information didn't improve your ability to diagnose individuals. And that's, again, because each one of these loci contributes very small additional increased risk. So that's the glass half empty side. And the question then is, where is the rest of this missing heritability to be found? One suggestion is that we just haven't looked hard enough. Let's keep going. Let's make larger and larger samples, and we will find more and more of these low impact variants. But models based on type 2 diabetes and other diseases suggest that this sort of asymptotes out at some point. So there are probably more loci to be found in this manner, but it's not likely to add up to the full amount, no matter how hard we try. And so attention in the field has really begun to focus on the idea that there are low-frequency variants of large effect. And that's schematized here, where we have the allele frequency of variants from essentially 0 to 10%, and whether they have a small impact on your risk of disease or a high impact. And so we start here with Mendelian forms of disease. These are like single gene. If you get this mutation, you will develop the disease. Those are rare and yet highly penetrant. GWAS, as I've said, Genomewide Association studies, they are able to find common variation that, because it's common, has a fairly low impact on risk. And what hasn't been able to be looked at until very recently is variation in this low frequency range that might have a sort of intermediate impact on risk and that could very quickly add up to the missing heritability. And that was the focus of the work that's presented in this paper, which I'm glad to be a part of, and which will be coming out in Nature a week from today, August 4th. So rush to the newsstand and buy it. And what we did here is a number of whole genome sequencing things, fairly modest and not very productive. And then we looked at what's called exome sequencing of 13,000 individuals. Exome sequencing is a shortcut where we just sequence the protein coding genes in a genome. And we looked then to see if we can find rare variation that hadn't been detected by GWAS. We looked in a large number of populations, globally diverse. I don't think I'll bore you with this, although for the population geneticist, this is the most exciting part of the presentation. This is also not particularly important, but we ended up with looking at almost 2 million variants in this study in 13,000 individuals. So large numbers now, because we are looking at rare variation. Basically any variant that even one person has will be detected using this approach. And here we plot our p-values for each of the chromosomes, and boom, just one little dot. One tiny result out of all that work. And it's actually an amino acid changing polymorphism, so it changes the sequence of a protein called PAX4. It's a fairly common allele, but only in one population, 10% frequency, and it's actually in East Asians, and conferring a fairly high risk. So it nearly doubles the risk of diabetes for these 10% of East Asians who have this allele and this gene. So maybe this, I was talking about the Asian paradox a moment ago and sort of dismissed the idea that there would be big allele frequency differences between genes and populations, and that's generally true, and maybe this is the one exception. And this actually may for some individuals, be in Asia, be a fairly significant risk factor for type 2 diabetes. It might even be rising to a predictive diagnostic level. It's a transcription factor that's integral in beta cell function and so on. It makes a lot of sense. It had actually been known because other mutations in the same gene did cause strict Mendelian forms of diabetes, but they were very rare in the population. So this is quite an unusual event. But the bottom line is that we find very little additional variation throughout the genome, which was a bit of a surprise and a disappointment. And this is true even when we enlarge the sample. And let me just show you these results here, which are consistent with the model that I showed you earlier. So this plots by allele frequency, going from rare, 0, to common, and shows the risk, the odds ratio of the alleles. And so it shows that for common variation, these hover just slightly above 1. And they do increase as the frequency goes down. And that's kind of what we were banking on and expecting. But because they're rare, we don't push them to sort of statistical significance, which would need really high levels of odds ratios or of allele frequency. So the trend is in the right direction, but we can't definitively say that any of these are causally related to diabetes, even though they might be. And that may just be an issue of sample size. So a summary here is that we don't think that rare variation so far is likely to add up to much of the remaining missing heritability. In Europeans, it only adds about 3% of additional risk, so we may go from 6% to 9%. We still have 20% of the genetic risk to explain. Though it does suggest that non-coding variation is likely to be important. So what we've looked at so far is only coding variation that changes protein sequence. So this has implications for this idea of personalized medicine and precision medicine. It was hoped that genetics would enable us to identify unique aspects of disease, unique to even individuals. We would optimize therapy and make a very personalized treatment program for particular patients based on the risks that we could predict for them using genetic results alone. And this, because of for the reasons I've explained, isn't likely to be the outcome. The variation is too common and has too small an impact on disease risk to make that useful or possible. Instead, I think we're gaining novel biological insights that will produce new therapeutic targets and new drug therapies in the future. And so the shift in the terminology you may have noticed went from personalized medicine, and now they're calling it precision medicine. So maybe we will more precisely target therapies to broad classes of patients, but not enter into this world where everyone is getting a unique, personally tailored therapeutic approach based just on their genotypes. So now I think, so we haven't explained all of genetic variation. I think we need to look elsewhere, and that's where epigenetics and the microbiome become interesting. And there's a number of new papers that are highlighting both aspects of epigenetics that might be related to famine and food abundance and scarcity in humans and metabolism, as well as these metagenome or microbiome studies as well. So one of the surprising observations epidemiologically from populations that have experienced famine is that often the offspring who are born to mothers pregnant during times of extreme famine are at increased risk of diabetes and obesity later in life, which is kind of a surprising observation when you think about it. And this has been called the thrifty phenotype hypothesis. So it's as if the baby is being programmed, as being, it senses from the environment that this is a time of food stress and food scarcity. And it's as if the baby is being prepared for that, and its metabolism is being adjusted for that possibility. And as it's born and the famine ends and it's born into a period of food abundance, that actually leads to a lifetime risk of obesity and type 2 diabetes. And there are a number of studies now that have looked at this. One was in the Ukraine in a famine in the early 1930s, where various provinces of Ukraine were heavily affected or moderately affected and less affected. And that showed that children who were born who were in gestation in these various periods, in the most severely affected regions, were always more likely to develop type 2 diabetes. And that sort of shifted along with the severity of caloric restriction in the parents. There's good data from China during the Great Leap Forward and famines there. And then also in the Netherlands in World War II. At the end of World War II, a sort of chaos had erupted and the Nazi occupation had not yet ended. There was a very severe famine there, and the offspring now who are in their 50s and 60s are affected. So how could this work? That's led to interest in what's called epigenetics. This is a fairly bizarre and newly understood phenomenon where aspects of the environment can lead to chemical modification of the DNA molecule, chiefly the addition of what's called a methyl group, a carbon with 3 hydrogens that gets covalently bound between C and G residues in the DNA. And those C and G residues usually precede the start of a gene. And when they're methylated, when there's a methyl group on them, it turns the gene off, essentially. It restricts the ability of the proteins that are going to copy the DNA into RNA and make protein. It prevents them from binding, shuts them off. There are other kinds of modifications as well, histone modifications that affect how open the chromosomes are and how accessible they are to the RNA polymerase as well. But the bottom line is that these chemical modifications can be affected by things like stress, environment, toxins. And so these methylation patterns could be the means by which a baby is reprogrammed and is without any change in genetics. Their genetic program is nevertheless altered in a way that prepares them for a period of metabolic stress. And again, that's shown here. You can have a metabolically stressed gestational period and end up with an obese child if it's born into, if the famine ends and it's born into a period of food abundance. And so there's only tantalizing evidence of this at the mechanistic level so far. And none of this work, by the way, is anything that I've been involved in. But just to show sort of proof of principle, this looks at the methylation patterns around a gene called insulin-like growth factor 2 in children of this Dutch hunger winter. So from December of 1944 through the spring. There was severe caloric restriction, rations as low as 500 calories a day, and only getting better as the Nazis lost the war and things came back to normal. And what it shows here is that compared to siblings who were born outside of the famine, there's less methylation in the children born during times of famine than those who were in gestation later in that famine. And that suggests that the gene activity of insulin-like growth factor would be higher in those individuals. And so it's at this point sort of anecdotal, but it is very consistent with this idea of thrifty phenotype, that a child can receive an environmental signal to adapt to a changed metabolic state through the readout of its genome through something like epigenetics. So then in the last part of the talk, I'll talk a bit about the gut microbiome. And this is equally fascinating and bizarre, and until recently not very well appreciated. And so, our body is made of 10 trillion cells, roughly. But there are 100 trillion bacterial cells that live within and on us. And so bacteria, of course, are much smaller than our cells. That's why they're able to outnumber us to such an extent. But, 2 to 3 kilograms of our own body weight are bacteria that are commensal. They're actually doing good things for us, or certainly not harming us in any way, even though our typical image of a bacterium is as an infectious pathogen that needs to be treated with antibiotics and eliminated. These things do very good things for us. And in a strange way, we're actually outnumbered by these bacteria. So they outnumber us 10 to 1 by cells, and the genes, because the number of species is so large, outnumber us by 50 to 1. There are more than 1 million microbial genes that are actively transcribed and expressed right within our own bodies. And thank goodness they are, because they help us digest food. And so you can imagine that the efficiency with which they break down polysaccharides or things that we can't normally digest could affect our metabolism. So bacteria that break down more polysaccharides and release more glucose into our bloodstream could be contributing to our obesity, and those that sequester more of that energy for themselves or don't break down those products could lead to a less obesogenic or diabetic risk. So that's the basic concept. Not a lot of very convincing data yet for type 2 diabetes. This is one early study that was published in Nature that shows a slight reduction in diversity in bacterial species within our gut for type 2 diabetics, but wasn't able to go much further than that. But in the context of reduced diversity, maybe those bacteria are metabolically active. I think the most convincing evidence so far is actually in the context of obesity. So it turns out if you look at the bacterial composition of a fat mouse versus a lean mouse, you see that the fat mouse has a reduction in this family called Bacteroidetes and more in this family called Firmicutes. And whereas the lean mouse, it's more even. If you take people and you put them on you take obese people, first of all, you take obese people and compare them with lean people, you see the lean people have more of this Bacteroidetes, just like the mice. And if you put them on a diet for a year and they lose weight, the Bacteroidetes abundance improves. So that's a fairly convincing piece of data. And in mice, if you do something fairly disgusting sounding, but do what's called a fecal transplant from an obese mouse into initially lean mouse, and from a lean mouse into a lean mouse, and feed them exactly the same thing, then the mouse that received the microbiome from the obese mouse itself becomes obese, everything else being equal. And the lean one stays lean. So that's about as convincing as you can get. And so this is a very promising area. It's becoming appreciated how important early aspects of our in utero and first year of life are. Whether you were born by Caesarean delivery or vaginal birth can have a huge impact on your eventual gut microbiome, whether you were breastfed or fed formula, when solid food was introduced, what was your antibiotic exposure early in life, vaccination history, infection history, all of these things contribute to your diversity, and it can have long-lasting, long-ranging, still poorly understood health effects. Certainly things like asthma, allergy, autoimmune disease, these things have very convincing links to the effects of early microbiome composition. The other hope is that, of course, that this could lead to interventions. If it's as simple as a fecal transplant, then maybe you can cultivate the right sort of healthy or protective microbiome that will keep you lean or not diabetic. And you can do that by probiotics, where, like yogurts or whatever, actual fermented food, where you introduce directly the microbes through diet, or what are called prebiotics, chemical nutrients, fibers, whatever, that encourage the growth of particular families of microbes. You could use targeted or intelligent use of antibiotics to eliminate particular species or of bacteria or families of bacteria that were harmful. And then ultimately you can do fecal transplantation, which is used in the treatment of various gut infections, for example. Clostridium difficile is currently used clinically with great efficacy to treat that illness. So, yeah, let me just end now very briefly with a discussion of a study. So that epigenetic and microbiome work was not my own, but it has stimulated us to look and try to design experiments that will allow us to disentangle the effects of genes and the environment. And one way we're doing this is with a collaboration in the Philippines. So as you may know, the United States actually ended up, at the same time that it seized Guantanamo Bay from Cuba, well, from the Spanish, and ended up with Guantanamo Bay and Puerto Rico, it actually ended up with the Philippines as well in 1899, and was sort of a colonial power until 1945. So there's a very large Filipino community in the United States, some going very far back, so long-standing community, and a lot of recent migrants as well. And so that enables us to look at people who are genetically similar but living in pretty radically different environments. If we can compare people living in the Philippines today with their genetic counterparts living in the different, very much more obesogenic environment of the United States, and begin to look at what is the impact of genetics versus the environment and things like the microbiome. So we're now currently embarking on a study where we enroll diabetics and healthy controls in Manila and the San Francisco Bay Area where I live. This shows by county in the United States the density of Filipino residents in the San Francisco Bay Area is one of the most popular areas in which they live, also Southern California. So it's actually, there are large numbers of immigrant individuals here with whom to do this kind of work. And so what we're planning to do is both to look at methylation in diabetics and controls in both of these two places and see if we see differences that we can correlate with aspects of the diet and the environment. And we're going to do a microbiome study as well. And we think this is a very powerful means of identifying and disentangling the effects of inheritance and the environment on things like complex disease risk. So we'll see. But hopefully we can add some substance to what are sort of tantalizing clues as yet, but not really proven associations with epigenetic variation and microbiome composition on aspects of metabolism. And just to end, I think Malaysia has some of the same possibilities, and I think it might be worth thinking about what unique aspects of, what unique contributions Malaysia could potentially make to this kind of research or other kinds of research, even if you think you might be sort of geographically isolated from a lot of the other, a lot of the scientific powers, or if you're dismayed by the amount of government funding you have, which, by the way, is universal globally. There are maybe things that you could take advantage of from your location here that I think could be very clever. And in fact, this is something that Maud has done, has pioneered quite a lot with her studies of population genetic variation of the Orang Asli populations. And she's actually made quite an international name for herself by studying those populations because they are so critical in showing insights into the early settling of Eurasia. But you could ask in Malaysia, because you have large communities of diverse individuals deriving ultimately from the Indian subcontinent, China, as well as indigenous Malay populations, living essentially in the same environment. So you have similar environment, dietary environment. There really is a nice Malaysian diet, even if the ethnic communities are a little bit enclaves unto themselves. There is sort of a common dietary and certainly physical environment to Malaysia. And so you could look at different genetic diversity in the same environment. That's very powerful. You could compare these immigrant populations to their ancestral populations in India or China, or in fact the large Chinese diaspora all over the world, and ask what is the effect of differing environments on these same genetic populations. Within the Malay populations and across Indonesia, to the extent that there are genetic connections and migratory connections, and between East and West Malaysia, I think there's a lot of potential there. And then the Orang Asli populations, which were until very recently hunter-gatherer populations. And so what is that transition like to go from hunter-gatherers, where you're basically under sort of chronic food scarcity permanently? What happens when you modernize in 1 or 2 generations and you're now living in an environment of great food abundance and overabundance? And so those are just some ideas that where I think you actually have a lot of competitive advantages or a lot of interesting things to do. And I mean these sort of as natural experiments, not to, these are human beings. I'm not suggesting anything disrespectful, but they are, from my vantage, experiments of nature that you could look at. So with that, I'd like to thank you for your attention and be happy to answer any questions you might have.
All right, so my name is Sadiq Rahman. I'm in the School of Science. So you gave a very, very detailed overview of the genetic background. But does that explain the rapid rise? I mean, I think that's a burning question. Why is there this almost doubling in 10 years?
Right. Well, so is this on? Well, so you're right. That's obviously not due to changes in DNA sequence or genetics as we commonly think of it. So I think the most obvious thing is our changes in the environment. We're much more sedentary, and we consume too much unhealthy food, and that's really, I think, the main one of the main differences. And maybe things like epigenetics. Maybe there are then these chemical modifications that are also sort of amplifying the effects of these environmental.
But the change is almost doubling in 10 years. Surely we're not twice as lazy as 10 years ago.
Right. Well, you might be. We might be. So things like the microbiome, for example, if that turns out to be significant, there's much more wide use of antibiotics, for example, especially early in childhood, and maybe that, and much more Caesarean delivery. Maybe many more children are being born with a depauperate or a different microbial set point than we understand. I don't know. There are in Asia in particular, the high prevalence of hepatitis B, because it affects the liver, and the liver is such an important metabolic organ in the glucose metabolism. That could be part of it. Yeah. Added sugar. The way even fruit juice, the way we drink fruit juice today is not the same as it would have been when people were actually, when it was much less frequent, and people weren't adding sugar or even consuming large volumes of fruit juice, and let alone soda and soft drinks and things like that. I don't know, Faisal, I don't know if you have insights too. I mean, it's your data that documents this, I think, most strikingly. And
Could we hear from Dr. Faisal?
Oh, thank you, Mark, for putting me on the spot.
Well, if you like.
I suppose there's many theories abound, and it's not being helped that we lack data really, especially on diet. Level of physical activity aside, I think diet, certainly in this population, play a major role. I think people don't realize what they're taking in right now in terms of, if you talk about, well, there are many aspects, right? Saturated fat, the types of fat that you're taking in, the types of carbohydrate you're taking in, frequency, timing.
And total calories, too.
And total calories, too. So, yeah, I can't say for certain, but I certainly agree the increase is tremendous. Compared to your data, we have a more recent survey, which we have repeated the same survey, population-based survey, in 2015. So it just keeps going up. I mean, this doesn't seem to be stopping yet for the prevalence of diabetes, both diagnosed and undiagnosed. So, yeah, so I can't say for certain as well what's the main contributing factors.
Thank you, Dr. Faisal. Dr. Seeley, behind there, in the middle. Yeah.
Good evening. Thank you very much, Prof. Mark, for your very interesting and educational lectures, and also Prof. Phip to give me information about this lecture. So that's why I try to make it today. I'm sorry, I'm a gynecologist, obstetrician from associate professor. So I do teach, and then I see patients, and then I take care of pregnant women and also the gynae patients as well. So it's alarming that what from our data is from our hospital, in a university hospital, is that 11% of women are now diabetic during the pregnancy. So we call it a gestational diabetic. So that is one of the alarming factors too. So it's as you've shown in the complications, the consequences of the diabetic in the women that, like the diabetic mother or gestational diabetic mother, and their baby tends to be big. So it looks chubby, but it's very difficult to deliver. So it's like at the time of delivery also a lot of complications and have to go for the cesarean section. And also you show that the baby the mother was diabetic, and in their life also have become a diabetic. So that is as one of the factors. Yeah, we usually have to take it seriously. And second thing is that it is very sadly and recently what I'm seeing is that a younger age of 28, age of 30, before they get married, they have this diabetic and they have endometrial cancer. So it's very sad news. We have no choice to remove their uterus even before. So it's like someone who had it age of 30 with cancer that is very heart-seeking phenomena. So we really have to go in to reduce this. I'm really glad about this genetic and looking for your, genetic things. But in Malaysia, what we found is that, like Dr. Faisal mentioned, the lifestyle, the food, for example, is like a buffet, you eat as much as you can, and we have 6 meals, and then, so it's like all this, I think we should start by now, because it's like if we wait for another 10 years, that will be worse than this. So I really want to join Prof. Mov and then also Dr. Faisal said if you have any campaign that, we have to bring down this diabetic in this part of the world, not only in Malaysia, Singapore, or this part of the region, I would like to volunteer and join you. Yeah, thank you.
Yes, thank you, Dr. Seeley, for your insight and sharing your experiences with us. Are there any questions or comments? Yes, please, at the back.
Hi, Prof. Mark. Thank you very much for your talk. It's very interesting to know about that. So just now you did mention that nowadays people too focus...
Sorry, can you identify yourself, please?
I'm Julie. I'm Julie.
And where are you from, Julie?
I'm actually from Sanofi, but I was a Monash alumni before that.
Thank you.
Okay, so my question is that you said currently the current medications... in terms of the medications wise, we are actually moving towards the precision medication, whereby in terms of, let's say, for example, sulfonylurea, you are focusing on certain genes, whereas the PPARG, you are focusing on, the other drugs are focusing on PPARG. So, but what we've known so far is that in terms of these two types of medications that has been given, it has been given as an oral medication, and it hasn't been able to reduce the blood glucose as effective as a basal insulin does. So my concern is, has there been any research that focuses on a certain gene that has shown that it is able to reduce the blood glucose to a very effective level, other than basal insulin?
Yeah, it's not exactly my expertise, but there are these glucose excretion genes in the kidney, and I guess there are those GLUT genes, or there are genes there that I think are leading to more excretion through the kidney, and that those seem to be working. What I was suggesting is that, not specifically about sulfonylureas and rosiglitazone, but the fact that their mechanism of action either reduces or amplifies the activity of certain genes suggests to me that if you tried doing that with all of the other genes now that have been identified as involved in the diabetes process through the existence of genetic variation that either increases or decreases your risk, you might end up finding, that might actually work. You might end up with more effective therapies that are more specific in having therapeutic efficacy. I do think it's a trove that pharmaceutical companies like yours, I guess, really should take very seriously, and some are finally. There's been a lot of reluctance. I told you that the effect of these mutations or these gene variants isn't very large, and so I think people have equated that with not being important, but that's really not the message. I mean, the fact that gene, if the effect of the mutation was too big, then it probably would be lethal, or it would disappear in the population. So the genetic variation is just a way of identifying genes that are important in the physiological process. And then if you turn them down, or turn them off, or turn them on, or somehow modulate their behavior, you might have a very big impact on you might have a very good treatment response that has nothing to do with any individual genetic or anyone's genotype or DNA sequence in those genes at all. Yeah. So I think that is gradually dawning on pharmaceutical companies, because they have had such poor success with the animal models. The other thing is, it turns out for a lot of these genetic results, we don't actually know exactly what's going on yet. So there's a lot of groundwork that still needs to be done to really flesh out for each of these 120 signals exactly which gene it is, and whether it's whether there's too much of it or too little, basically, things like that. And pharmaceutical companies are reluctant to invest big time until that's better understood, given it takes 10 years to get these things developed and through clinical trials and all the rest of it, so it's a very expensive process. But I do think this will end up being more precise, and there will be less drug failures along the way if we take advantage of this information.
Yeah, just to add on, I think I certainly concur with Mark, and the thing is that a single gene doesn't act alone, and oftentimes it's acting in concert in a network of other genes and the cellular environment. So although you're targeting it, it might not be precisely that you're targeting. So getting, as he mentions, maybe a constellation of a few genes acting together might sort of do the trick better within a particular background of a cell or something. So it's very rare that you get a single gene, single action, single drug. It does work for certain things, but in complex disease such as diabetes, it's going to be very quite a challenge, I would imagine. Yeah.
Thank you. And there is one more question regarding to the GWAS results, right? The GWAS studies. What sort of samples do they actually use to collect?
Well, those are usually just patients, people who are diagnosed type 2 diabetes, and you just take a DNA sample and then find healthy people without diabetes who are the same age and gender and all the rest of it from the same population. And then, the problem is you need large numbers. So these studies have now involved over 100,000 diabetics and 100 or 200,000 controls. And that gives you the statistical power you need to find what are pretty subtle effects when you're looking across something as big as the whole human genome. But a lot of it varies. So some studies are just self-declared. People say they've been diagnosed with diabetes, and so you consider them diabetics. And of course, with the controls, you never know if they might go on to develop diabetes or not, or you might have just taken their self-report that they're not diabetic and not done a full glucose challenge test or things like that.
Yes, Dr. Sile again, please. Students, you better think of some questions to ask. This is your chance.
Yeah. So thank you very much. I have read your Singapore study because it's like a really impressive, like with the 8,000 patients that collected for the looking at the loci transferring the loci. So it's like in your study that you have the Chinese population and Malay and Indian, like which reflects our Malaysian population as well. Do you have any kind of a mutation, gene mutation, when they have sort of a cross marriage? Yeah, right. For example, Chinese married with the Indian and their descendants. Right. Yeah. Thank you.
Well, I mean, you see that. You can see that genetically very clearly. And usually what we do is we use a statistical method to correct for that. But I don't think you're asking if there are combinations of genes that come from one population and come together in one individual, whereas they had never been seen in the same individual before. That's a very exciting possibility that even without intermarriage could exist, and that's a very difficult question to get at, because you're asking if two different genetic variants together have a bigger impact than either one does alone, or the sum of the two, and that's there's a lot of interest in that intellectually, but it turns out to be it's hard to do that properly statistically in an efficient way. It ends up being, you end up testing so many different hypotheses that you need astronomical levels of significance. So what people have done is taken just a limited number of genes that have been identified through GWAS, say those 100 genes, and then look at pairwise interactions amongst those. And that hasn't been very productive. So it was a bit of a surprise. The thinking was that you would find in two genes together might have a bigger risk overall than either one did alone, or the sum of the two. And so far that hasn't shown up much promise. But I don't think that means it doesn't exist. It's just very hard with our current methods to find that.
Any comments? Any questions? Yeah, you showed a slide. It was quickly taken off about the gut microbiota, Mark. And could you sort of maybe explain to us, for my understanding and for maybe people in the audience, about that metformin connection? Because we know that metformin is a very common drug that is used to treat diabetes. So what's the gut microbiota and got to do with metformin?
Yeah, so there was an initial study on type 2 diabetes that didn't account for the therapies that people were on. And it did show an effect of diabetes did show differences in bacterial compositions between diabetics and healthy people. But people have looked subsequently and think that a lot of that does have to do with metformin therapy, which is the most common sort of first-line drug used to treat diabetes. So it may be that what has been seen so far is more a response to the metformin treatment than to diabetes alone. That's going to be the tricky thing, I think, is to determine causality. Maybe there are different microbiome compositions coincidentally, or that relate to the metabolic dysbiosis that occurs, and that it's not that the bacteria are actually driving the disease, they're just sort of reflecting of the disease process. Now for obesity, I think the evidence is a little more convincing, and when you can do the transplantation of microbiome in mice and see the effect go along with that, I think that's pretty convincing evidence, and maybe the same possibilities will arise with type 2 diabetes, but determining causality is so far been tricky.
Yes, as a student there, yes. Introduce yourself, please.
Hello, I'm Adrian from Year 1 MBBS student of Monash, Malaysia. Sorry. We do know that there is a number of rising population of pre-diabetes that is happening around the world. And there are also a number of population who is diagnosed as pre-diabetes, but they are neither obese or overweight. So are there any research done on this part? Is there any genetic variation that causes this to happen?
There's some. So there's a form of diabetes called maturity onset diabetes of the young, and that is a single gene. That tends to be a single gene, single mutation disorder that occurs independent of obesity or really typical risk factors. But that's rare. So I think prediabetes is certainly of interest. There have been associations, there have been GWAS studies of insulin levels or glucose levels as well, just as a trait. You could see what genetic variants influence glucose levels, which is really how prediabetes is measured, is just by a certain fasting glucose level. And those have identified different genes that are not associated with frank diabetes, but that do somehow do elevate the levels of glucose. So that is certainly interesting. I think, other than that, I don't know what to say. The good news is prediabetes is reversible. The other thing I didn't mention is that gastric bypass surgery is actually pretty effective in very quickly reversing type 2 diabetes. And there is some thought that that's through an effect on the microbiome composition as well, which does shift pretty drastically. And so that is increasingly being indicated in the US at least for overweight type 2 diabetics. And that can work so almost, miraculously, at least for a while.
So sure about that. What does it entail, this gastric bypass surgery? How much do they remove?
Oh, I'm not really an expert on that, but it's you reduce the volume with balloons or bands or something that restricts the size of the stomach basically, and
So you're eating less then.
You should be eating less,
weight.
but humans being humans, some people find ways around that. In the back.
Was there somebody behind? Yes.
Okay, hi. My name is Navina. I'm a pharmacist, but currently a postgrad student here. So I have a question based on, like, all the individuals who have diabetes. They have various different reasons, as in what mechanisms of action that is malfunctioning in their bodies. So basically, it's either a reduce in insulin production or an increase in insulin resistance. So that's the main factors. And currently, since there are so many different reasons as to why they have diabetes, is it a single factor that will unite all these different differences in their problems? Is there a way into finding that, and will that help in diagnostic procedures and also in terms of like medication? Because as we all know, there are different classes of medication, and not each class is effective for each individual. Thank you.
Yeah, that's certainly been the hope. But I think it's so far been a little bit disappointing. That's one reason it actually matters whether the variation is common in the population or rare. The field has shifted very rapidly to try to look for rare variation by doing whole genome sequencing, with the idea being that maybe it is. Everyone has diabetes for their own unique reason, or their family, or their close relatives, and so that would be important to know. It might be disheartening. I mean, if every diabetic was diabetic for a different reason, that might throw up real obstacles to, you're not going to develop a million different therapies for a million different types of diabetes. And so far, there isn't much evidence in support of rare variation being, the single cause of diabetes. So that extreme doesn't look like it's going to occur. So, and that might be a good thing. So, maybe there are 3 or 4 subtypes of diabetes we can end up identifying using genetics, and maybe we can suggest particular therapies. That's certainly true for MODY, those maturity onset diabetes of the young. Knowing which gene is mutated and which mutation it is can immediately suggest therapies that will work and those that will definitely not work. And so if you extend that to the more common type 2 diabetes, you might end up in that realm as well. But so far, there isn't much evidence of that.
here in front, second row, to the third row up there. Yeah.
Hi, my name's Li Fang. I'm a Monash student here. So I'm not sure if you know about the Baba Nyonya community in Malaysia.
Oh, I know very well. We were just discussing the Bua Kelawak.
So
How
like,
about the Thai as well? Tomorrow. Okay.
so like some of my family members, they are from the Baba Nyonya community, and like this is an informal observation, because I have no data to support whatever I'm saying, but their family, they eat very high sugar desserts and stuff like that. So the food that they eat are very high sugar intake, but it's very rare that they see family members with diabetes. So could it be connected to their genes, or like what could be because they've exposed to high sugar since young?
It could be. First thing is, I mean, it's great to make observations like that. I mean, that's how science is done, right? You make an observation and form a hypothesis. And so now the question is, how do you test it? And I don't really know the answer to that, but that's certainly the right impulse. Yeah, it's really hard to say. There are so many. I mean, coffee consumption actually is protective for type 2 diabetes. I don't know if
Compelling.
so the possibilities are endless.
Yeah.
There may be something to that observation, and it may suggest a kind of epidemiological study that someone could perform. That's kind of related to the question earlier about what happens, when you have admixed populations, and you have, are there risk and protective things that end up coming together in a way that is unique? Who knows?
Well,
Yeah,
the challenging thing is always the numbers, isn't it? It's a numbers game. I mean, you saw those numbers, hundreds of thousands, tens of thousands. So if you want to go sort of a genetic direction, then the numbers really have to come out, and it has to be of statistical significance. So there's always the challenge of the numbers. How many to be believable, how many cohorts you're going to look at, and what sort of, measurements you're going to do. Another question, Lifa?
No, that's it. Thank you.
Okay.
Thanks.
Hello, my name is Dr. Loh. I work for an insurance company. I've started people on metformin. Of course, the old idea, metformin helps sugar to enter the muscle. But you're postulating that metformin actually changes the gut bacteria. So many patients when we start, they have the diarrhea. So after a while, the diarrhea goes up. So is there a possibility there's a change in the gut bacteria causing, helping in the diabetes and bringing down the blood sugar? Thank you.
Yeah, no, that's another great observation. I don't know, and maybe that does suggest that there is, that would certainly be worth looking at.
Hi, my name is Crystal. I'm a first-year medical student, and we were thinking that there was a research done that when mothers are in famine, their offsprings have the tendency to have type 2 diabetes. What about if their mothers have type 2 diabetes? Would their offsprings be healthy due to the epigenetic thing?
Yes, I'm not sure. That sort of relates to the other question, and she was saying that the birth weight is actually higher for the mothers of type 2 diabetes. And funny enough, high birth weight is not associated with type 2 diabetes. So that isn't, the child might end up being better off than you would expect, even if they start off heavier than, that doesn't necessarily predict they'll be heavier as adults or more prone to diabetes. So this may be known, and I simply don't know the answer, but I may be that's protective.
But then there's that something we know of where a person who develops gestational diabetes, the mother, and there's a tendency for her to have a larger child.
Yes,
that's another thing.
And but that large child may not go on to be obese or diabetic.
Yes.
interestingly enough. Now, the mother is at very high risk of getting type 2 diabetes soon thereafter. I mean, that's pretty well understood. Gestational diabetes is a great predictor of later type 2 diabetes development.
Thank you very much, Mark. Hi, I'm BP from UCSI University. Continuing on the questions about the mothers deliver babies with overweight, and I'm just wondering because there has been a study, if correct me if I'm wrong, but there was a study that I came across that they did a study on the gut microbiome of the mothers and the baby. They actually found that there's actually inherited patterns of the gut microbiome patterns. Well, I'm wondering if that could be a possibility of.
Oh, I see. Yeah. No, the mother is really the key. I mean, the natural microbiome is really dependent on the mother. Except if it's by cesarean delivery, then those children have a very distinct gut microbiome, which is more characteristic of the bacteria you find on the surface of the skin, not so much within internally.
Yeah. So there's another question. It's actually quick questions. And do you still believe the thrifty gene hypothesis in diabetes?
Thrifty gene. Well, that's something I didn't talk about, and so that's. Gosh, I wonder if I can even. So that's the idea that. Through severe selection, right? So maybe these Pacific islands where people sailed in a canoe for 2 weeks into the unknown and ended up on a desert island, that enough people died in that kind of extreme scenario that the only ones who survived were the ones who were so metabolically efficient that they could survive those kinds of voyages, and then therefore. The descendants are all at extreme risk for type 2 diabetes. I guess I do kind of accept that as possible. I mean, certainly those Pacific Island populations are universally high prevalence of diabetes and have been for a very long time. I think there was even a paper just a couple of weeks ago on Samoans or one of the Pacific Island populations where there was a distinct genetic variant of pretty high prevalence. I think in Nature Genetics, maybe we should look either on the Advanced Online or within the last couple of issues, there might be some insight in that.
I think we'll have.
There's one more.
Okay, 2 more questions, and then we will call it a night. There's a lady there.
My name is Aruna. I'm a medical officer with the Ministry of Health. So going back to your slide about precision and personalized medicine.
Right.
So being in MOH, a lot of our targets are based on clinical outcomes of patients. So what I gather from your lecture about the whole effects of genetics on clinical outcomes of diabetic patients is so obviously it's more of an environment thing than a genetic contribution, right? So what's your opinion on personalized genetic-based medicine as compared to treatment based on perhaps novel indicators in cost effectiveness aside? We're not talking about cost in the general population. Thank you.
Right. Well, I'm a geneticist, and I sort of bet my career that genetics was going to be the thing. But oddly enough, I don't believe it. I'm almost glad that it's not necessarily turning out that way. So that I don't think we will end up with 1,000 different therapeutic approaches to 1,000 different genetic types of people or disease. That's probably in the long run a good thing. So I think, I really do think the power of genetics is it will just give us more targets to modulate, and some of which will have a beneficial impact on disease indicators, regardless of what an individual's genotype might be. Hopefully, that's the scenario. So we end up with a scenario where there might be 10 or 20 different drugs that would all be fairly effective in most individuals, and not necessarily worrying about tailoring them to specific types. Now, the counterexample, as I was suggesting before, is MODY. So there are specific therapies that you really can know are going to work pretty well, or know that aren't going to be worth trying. And I guess the scary possibility is that type 2 diabetes would end up like that. But genetically, those are very different things. I mean, type 2 diabetes is the constellation of many different genes of pretty minor impact individually, and together with the environment. Whereas MODY is pretty much one gene, you're definitely going to get the disease.
But certainly we would agree, I think, that there's so much that can be done in terms of lifestyle changes and how to prevent these pre-diabetics from actually moving to the diabetic spectrum. That I think is going to be major.
Well, yes, but you and I were talking this morning about as a health education. I'm forgetting your name, but you were saying it's very difficult to get changes in Priya. Yeah, it's very difficult to get those changes in diet and lifestyle, even though we all know how important they are, myself included.
Yeah, so now we've got more tax on sugar to stop us from consuming too much teh tarik and things like that. Just to round off, are there any more questions, please? One more before we wrap up. Farhang, yes, he's been wanting to ask for a long time.
Hi, my name is Farhang, PhD student in the School of Medicine. My question was regarding the GWAS studies, because GWAS studies usually focus on coding area. So how about non-coding area? For example, based on my experience, when we do some analysis on selection, it's not actually.
Well, so actually, GWAS was not really centered on coding variation, and if you look carefully, it turns out that many of them are not mapping to coding genes, and so when we did the exome sequencing approach, we did it for economic and technological reasons. And I would have preferred to do whole genome sequencing so we would have the whole genome and be able to look at those intergenic regions. So I think, if you want to really go after this rare variant hypothesis, that's where you look next. The problem is it's just so hard to interpret. If you find something in a non-coding sequence, your ability to even guess what might be going on is almost zero. But that's what's exciting about science, right? That's the challenge. That's the new frontier. And hopefully some of the young people in this crowd will take up that challenge.
Okay, I think thank you very much for your questions and your comments and being so interactive. I think it just falls to me to once again thank you and also thank Professor Seielstad for his talk. And we are now going to present a token of appreciation. And we get Professor Mahindran Nair, the DPVC Research, to actually do the honors.